This video will go over the basics of the microscope included in the CyanOScope kit. The kit comes with a student-grade microscope so that you can record your findings when you collect bloom samples. The stage of the microscope is where you place your prepared slide for observation. The dial on the side of the microscope controls the vertical movement of the stage. The outer dial makes larger adjustments to the stage, while the smaller inner dial is used to make fine adjustments. A mechanical stage has been added to the microscope to allow you to control the slide's position on the stage laterally as well as forwards and backwards. The microscope has three main objectives: 4x, which is the smallest and is marked in red, 10x marked in yellow, and 40x, which is the largest and marked in blue. The top ocular objective has a 10x magnification, allowing your view to range from 40x to 400x. The ocular objective slides in and out of the microscope in order to allow you to insert the digital eyepiece that connects to your computer, which we'll go over later in the video. The microscope comes with a backlight that can be powered by batteries or by plugging it into an outlet using the included AC adapter. The battery compartment is found on the base of the microscope and can be opened using an Allen wrench that comes with the kit. The AC jack is located on the back of the microscope next to the backlight's on-off switch. Different microscopes have different ways of controlling the brightness of the backlight. Some models have a dimmer wheel located on the side of the microscope, while others have an aperture wheel attached to the underside of the stage. Turning the aperture wheel changes the amount of light on the slide. The kit also comes with a small set of glass slides and cover slips, as well as a counting cell and a calibration slide. These are delicate and can be sharp, so make sure you are careful while handling the slides. To assemble your slide, use a pipette to gather some of your collected sample and place a drop onto the slide, and cover with a cover slip. With the cover slip in place, carefully place your prepared slide onto the stage using the clip from the mechanical stage to secure it. Always begin focusing using the smallest objective to make sure you don't accidentally crack the slide. The smallest objective in our case is 4x. Use the dials on the side of the microscope and on the mechanical stage to position your sample and bring it into focus. After the slide is in focus, you can remove the top ocular objective and replace it with the digital eyepiece. When you switch the eyepieces, make sure you use the protected covers to shield the glass from dirt, dust, and other particles that may harm the eyepiece. Now we will move on to the computer using the software included with the microscope kit. In the software, make sure the device selection matches the model of the microscope you are using. Go to the Video tab to see the live view of the slide. After the image appears in the software, you may need to make additional fine adjustments to focus the image. It's important to keep in mind that there is a delay between when adjustments are made on the microscope and seeing them on your computer, so be sure your adjustments at this point are small and give the software a moment to catch up so you can see how your adjustments affected the image. After the slide is framed and in focus, click the button that says Snap. It will capture and save the image. After clicking Snap, the image you just captured will be displayed as a tab in the software for you to check. You will have to select the Video tab again to capture your next image.